All right. Well, thanks everyone for coming to the talk. Yeah, I've wanted to do this talk about clinical performance assessments for a while. It's pretty relevant for a lot of devices that we handle, and especially for machine learning enabled devices that do some sort of detection. It's definitely relevant for those things. So what I'm going to do, I did prepare a few slides. Let me see if I can share that easily. One sec. Why isn't that working? Hey, David.
Yeah, how's it going? Hey, everyone.
If I try this, is that big screen showing up?
Yes.
Okay, it's working. Okay, yeah, I've wanted to talk about clinical performance assessments a while, and yeah, this is in the context of computer-assisted detection devices. So yeah, we'll start with what are clinical performance assessments, and then when are they required by the FDA, and then I want to highlight some of the details of clinical performance assessments, mostly just get a little bit more information about kind of the nuances of these studies, things that I've seen before, and yeah, maybe that'll help future submissions for anyone that's involved in a study like this. So yeah, what are clinical performance assessments? This is not like an official definition, but this is kind of a combination of multiple definitions that I found. The clinical performance assessment is an evaluation process designed to demonstrate the effectiveness of a device as it is used by the intended users on its intended patient population. And specifically for CADI devices, this type of assessment should demonstrate that when the readers use the device, that they improve in some way, typically something like their sensitivity at finding something, versus when they're not using the device. That's kind of the main goal of this type of evaluation, showing that your device has a positive effect, both on its intended patient population and the users that are using it. When are they needed? So just getting kind of in the weeds on the regulatory stuff, the FDA does say that for 21 CFR 892/2050, sometimes it is required, and then more often for 892/2070, it is required. And they do list out specific product codes where these are CADI devices that they are required, but there are additional ones, like an LLZ potentially, I guess, could require a clinical performance assessment. When your device has the potential to influence the interpretation of an image, that is likely when you'll need some sort of clinical performance assessments. It may not be designed exactly like they are for like a CADI device, but if you're influencing a reader in some way, there likely will need to be this type of assessment. You'll probably need it if your chosen predicate has this type of assessment. It'd be really hard to get away from that. And you definitely need it when the FDA tells you to. Yeah, they'll let you know if it's required during a pre-sub. And there may even be other reasons not covered here, but that's kind of the main ones that to look out for with a new device looking for clearance, something that might need this type of evaluation. So yeah, I'll get into some of the details of these studies. I hope everyone did get a chance to go through the kind of the template that I had created. That one was based on a previous submission for a client where we designed a MRMC study. And a lot of this is kind of based off of that. There are going to be lots of different ways to do these types of evaluations. There's no one size fits all. I hope the template is general enough that it could be tailored to any future projects. But yeah, I encourage you to comment on those pages so we can maybe come up with something that would be really useful. But yeah, one of the steps for this type of evaluation is to go out and collect data. It needs to be assessed on data that's included in the patient population, the intended patient population for your device. Typically, especially with CADI devices, it would contain disease cases and non-disease cases. And we'll get into the reason for that. You want to know sensitivity and specificity. So you'll need both types of patients in your data set. And when you're collecting data, it's good to early, as part of your protocol, to establish an exclusion and inclusion criteria to explain what types of patients will be included, certain situations that would exclude them from the study. And then, yeah, big thing for CADI devices is you must separate this data from any of the data that was part of your development or training of the device. Definitely for something like a machine learning based device, it cannot be associated with the training data. It's got to be completely separate. Once you have your data, there's need to establish the ground truth of what's inside that data. And this all depends on what your device does, types of things it's detecting, what it's, in this case, like segmenting. For this example here, you may have a device that one of its function is to segment normal anatomy, like an organ. Typically for normal anatomy and large anatomy, that it's a little bit easier of a task. So it's not something that is maybe a ton of variation associated with it. So this type of ground truth establishment, where you have three annotators just kind of independently doing their segmentations of the liver, and then a computer program that's actually an algorithm staple can be used to combine those three into a single segmentation, and then you can use that as your ground truth. That is one example of using it to establish a ground truth for something like a liver. For more difficult segmentation tasks like lesions, something that's not supposed to be there in the body, typically you'll need a better way to establish, this is the ground truth. I hope this shows up okay for everyone. It is also on that template page if you want like a zoomed in version. But because lesions they can have a lot of variability when you do the segmentations, depends on which reader is looking at it, depends on, like what type of evaluation software they're using. Like lots of factors can affect what the final segmentation of a lesion will look like. It's also the opinion of the reader. I hope my cursor is showing up okay. For annotator 1, they see one lesion, but for annotator 2, they actually see two lesions. And then annotator 3 might see one big lesion. What do you do about that? There's this variation in what they believe is the true segmentation. So there's no like surefire way to establish the ground truth. The FDA does accept, I think, multiple methods to do it. This is just one way to do it. Maybe a little bit on the more difficult side, but what you end up with is a very strong ground truth by doing it this way. The method here is that there would be an initial annotation. These would be annotators that are not top experts in their field. I would say they could even be annotators from outside the US, would produce the initial segmentation. That segmentation is given to a US radiologist that has a bit more experience that would approve the segmentation. And if there's any error of the segmentation, they would actually change it. And that's what I'm showing with this path right here. The reviewer changes it to two lesions. And then there would be a consolidation step where you would identify lesions that were annotated and also identify where there might be a discrepancy. That's what the yellow is showing, that reviewer 1 did not find this second lesion, so something's going on there. There needs to be an additional step to establish this ground truth. There needs to be like a tiebreaker. One way to do that would be, majority vote. These two reviewers saw this one didn't, so reviewer 1 is wrong, and then just accept the reviewer 2 and reviewer 3. What we often, at least what I've often seen, is that the FDA likes the use of an adjudicator, which is an additional expert that really just serves as the tiebreaker. They're presented with the lesions that have these discrepancies, and then they make the final decision. So that's what this is showing here. There's this yellow lesion that is kind of the suspect lesion. Then the adjudicator makes the final decision. Yep, this is a lesion and this is the boundary for it. It's kind of a really tedious part of the study, being able to go through all of your data. Like in some cases, you have hundreds of cases that need to be ground truthed. But in order to give evidence to the FDA that this is a strong ground truth that isn't suspect or subject to a lot of variability, this might be a method that you'll have to use to establish that. I know that was a lot kind of in this path, so I do want to pause if there's any questions on what we're talking about on this slide. Yeah, I'm open to any questions or if something's not clear.
You said when the adjudicator is given an MRI, an image that has a discrepancy, are they also given the opinions of the previous reviewers to take into account what others have said, or just?
Yeah, I think there's actually either way would work. You would just have to be part of your protocol. Like one way would be present them with the opinions of all three and say, hey, which one is correct, or give them this kind of combined version where they would say, just edit this annotation so that you believe it's correct. That would be another way to do it. Again, there's not like a cut and dry way to do these adjudication steps, or this entire protocol for ground truthing. This is just one way, and it's definitely recommended to propose these protocols in a pre-sub with the FDA so that they can look at it and say like, yep, that's a good way to do it. And then maybe sometimes if there's an easier way to do things, I don't know if the FDA would actually recommend that to you, but yeah, it's definitely good to propose your protocol in a pre-sub to have it looked at by the FDA before you go out and do it.
This isn't really a question, but maybe just a few comments that might be of interest. So there's almost always this tension between the cost of doing this, because of course you have these expert radiologists who you're paying a lot of money, and especially if you have a lot of cases. So there's the cost on the one hand, and then there's what the FDA will let you get through your submission on the other hand. And a lot of what we're doing is helping companies try to balance those two needs so that they don't go out of business because it costs so much, but they also don't go out of business because they can't get past the FDA. And like, man, there's so many places in this chart here where things have gone wrong. Like, we've done annotating with a separate AI model for the first step, and sometimes it's okay, sometimes it's not. There's also questions around the reviewer's expertise, like how many years of experience do they have? We've had FDA say, hey, these guys aren't experienced enough. We even had a case recently where they got a non-substantial equivalence where in the argument was, hey, even though we agree your readers are all good, they're so variable from each other that we just don't understand how to interpret the data, that one was a really surprising one. And we almost appealed that. And yeah, so there's a lot of ways. We've had questions about how you combine the three together. Like the staple method is one way, but there's other ways to do it. Sometimes the adjudicator is actually the third person, so you'll have two people do it, and then a third will only look at it. So there's a lot of different ways you can do it, and it definitely depends a lot on the specifics, clinical situation, and a lot of factors. But anyway,
Yeah.
sorry to jump in, Ethan, but just
no, that's all right. That's really a good point, because like, why even have this first step where there's these annotators that are then passed on to another set of three people? One of the reasons is sometimes this first step can take multiple hours. And like I said at the start, these could be radiologists maybe outside the US or from a annotation service that are maybe not necessarily experts, but they're cheaper than these reviewers that have 10 years of experience. So they put in the grunt work of the 2 hours of annotation for each case at a cheaper cost, and then hand it off to the reviewer, which maybe spends 15-30 minutes of time to just review the case. And so it's a cost savings to sometimes do it this way. That's another reason for that. Yeah, wanting to balance having a really strong ground truth versus not having to pay so much for achieving that ground truth. Yeah. Saman, did you have a question?
Yeah, I wanted to ask David, how do they determine whether or not like they're too variable? Are you saying like variable in their opinions on what the ground truth is, or in their like experience, or
too variable about their opinions of what the ground truth is exactly? And another factor, it's for non-standard things that if the radiologists aren't doing it in their day-to-day practice, and sometimes even if they do, like it's like, where exactly is the boundary of this lesion? Like, it's not always clear, and you could do it multiple different ways. And so having a protocol that's really detailed that all the radiologists actually read and actually follow is a big part of this too. And what can happen is you can have variability between the readers just because they define their boundaries a little differently. And we've had studies where that happens, and then the results aren't good. And then it's like, well, okay, can we go back and edit it? And like, there's all these kind of questions that come up. And another really common issue is if the training data was done using a kind of a looser protocol by less skilled people, and then the radiologists have their own opinions about how the annotations are done, then your performance may not be very good because the model is trained to do something a little bit different than what the reviewers do, and that's also problematic. But anyway, yeah.
Yeah, all good points.
I have a question on the reviewers. What has been your experience with the FDA? Do they all have to be US board certified?
I mean, I would say yes. That's what they really want.
That's what they
I've
want?
not been in a situation where they've accepted anyone outside the US for that role, like the step here, and definitely
Okay.
not for the adjudicator.
Okay. Thank you.
So I
From
also have a question. So from the figure, it seems like the data from annotator one all goes to reviewer one. Would you ever want to mix up some data between the different reviewers? Because if, let's say, the annotator and the reviewer have a similar bias, it might amplify it.
Yeah, that's actually something I thought about. It definitely complicates the diagram, but essentially annotator one for certain number of cases would be reviewed by reviewer two. Is that what you were saying, Ilya?
Yeah, basically give, mix and match, like, separate into three piles and then give everybody, annotations from three annotators to each reviewer.
Yeah.
I guess you don't want to duplicate the whole data set, but give them a subset of all the annotations.
Well, yeah, in this case, there's three instances of an annotation for one patient. But yeah, I definitely know what you mean. It would probably be best if there's just this initial step where the three annotators make their annotations and then the reviewers review a sample from each annotator. So that they're not just reviewing one annotator. I do believe that would be a better method. Maybe not necessary, but yeah, probably a better method to avoid certain biases, or, like you said, they might have a similar bias and that's always being propagated throughout the throughout the annotation protocol. So
would it be too expensive to go back and redo that if you find too much variability, or now that you're in the process, you can't change it?
For this situation, it might be time. That's the issue. For that scenario where you would be like crisscrossing the annotators with the reviewers, you'd probably need to have them go through the entire data set before you hand it off to the reviewers, because then you'd have to provide the sample to the reviewers. With this method, as soon as annotator one is done with a case, they hand it off to reviewer one. It's just a little bit more efficient that way.
Sorry.
Okay, thank you.
One more question, sorry. I was wondering, because you had said there's really no set way to do this process. Are there any guidelines, or is it just kind of that they want experts to review it?
I mean, there is the caddy, I'm forgetting the name of it. This one right here. I'm sorry, it's not on the screen. Computer-assisted detection devices applied to radiology images and radiology device data. That one does have like a little bit of guidance on establishing ground truth, but it's still pretty high level. It all depends on the domain, depends on the target, what you're actually trying to detect. Yeah, for certain types of lesions, maybe they're easy to segment and there's not that much variability across readers. So maybe a less strict protocol would be needed. Yeah, it's really variable. It's hard to know for sure. That's why doing a pre-sub and talking with the FDA about it before committing to a protocol is probably the best way.
So it is case specific.
Yeah.
Question.
Yeah.
Is there a way to have like a rationale? So if you're trying to generate, I guess, a bunch of reviews done where you have a situation where like the content is very niche or maybe doesn't have that many experts, is there a way, is it like a hard and fast rule that you need X amount of reviewers, or is there a way that you can give rationale of this is a really niche symptom or something, or disease for that matter, and you just don't have the resources available to actually have that many reviewers? You might have a lot of annotators, but you're like, your hands are tied for the amount of reviewers you can get.
Hey, yeah, that's definitely a not a good place to be in. For that situation, again, a pre-sub with the FDA to explain it to them is definitely needed. Like for the situation with it's brains of pediatric patients. Not a lot of radiologists are, qualified to do segmentations on those brains. You typically want a neuroradiologist, someone that's specific to the brain, to do that. So we kind of ran into that. They definitely wanted neuroradiologists to be the reviewers. So yeah, we kind of had to find those, and that wasn't very easy. But yeah, I'm not sure is my answer.
I can say there's another client that we'd worked on where we were able to make the case that only one reader was needed and not three. So I wouldn't say never, but I would say most of the time you do need three. It kind of depends on the intended use of the device and how important it is to assess the inter-reader variability. And even in that case, the way it was done was doing a study to show that the inter-reader variability was low because they were very standard and easy things to measure. So you had to justify why it wasn't appropriate or wasn't necessary to do all three readers in that case. So
yeah, thanks, David. Yeah, let's get into the actual study design. So you've went out and collected all your data. You've done the ground truthing part. The next step is to actually evaluate what your device's effect has on the readers that use it. So the way that is evaluated is a multiple reader, multiple case study, where each reader will interpret each case twice under different conditions, one using the device, one not using the device. And because it's done both ways, you can measure the effect that it has on those readers. For most situations, a washout period and a specific case reading order is used to remove or to reduce the potential for biases. One of those is memorization. Let me actually go to the next slide. So this is an example of the reading scenarios for memorization. So each reader has to read each case twice. You'll read, say in group A, you'll read case one in reading scenario one without assistance of the device. And then there'll be a 30-day break where you don't do anything. And then session two, you'll read that same case again, but have the assistance of the CAD device. And all of these will be in a random order. And the reason this is done is there's a potential that if you don't wait 30 days, the reader can, oh, I remember this case. I don't know how often that actually happens, but this 30-day washout period is supposed to, mitigate that effect. And then we've had a lot of feedback from the FDA about handling these reading scenarios. I have done a study where every single reader started without the device and then 30-day washout period, and then they use the device. But the FDA has been pushing back recently to have these types of scenarios where the day that you read the cases, half of them will be with the CAD device and half of them will be without. And doing it that way also mitigates certain effects of having the order of whether you're looking at it with the device or not. So yeah, that's kind of an interesting thing, and it is quite a bit of bookkeeping to set up a study like this, making sure that every reader is reading every case and that they're read in different orders, but they're having them read in different reading scenarios. So it is pretty complicated. I hope this diagram, kudos to Mary M for putting together, is helpful. The main idea is that you set up your study design so that readers go through the cases in a different order under different scenarios, that they will eventually go through every case once, and then there's a break of 30 days, and then they'll go through the cases again. And then every case will be, the reading scenario will be flipped on the second day. So,
Hey, Ethan.
go ahead, Mary.
Yeah, on the other slide. So I had it come up one time where the client really didn't want to do the first session half with CAD and half without CAD because they felt that just through use of the device that the readers would inherently get better without the device too, that the device itself and by using it with the AI that they would figure out how to do it better and that they would become more accurate just through using the tool, to where they tried to push back with the FDA and argue that it would falsely inflate the use without the device in the second session because they would have effectively been trained using the device first session, but the FDA didn't buy that argument, and they still wanted them to separate the session the way that you've drawn it out here. But I thought that was kind of interesting that they were pretty confident that their device was going to make people better even when they weren't using the tool. So it was interesting.
And I agree with that for the most part. Like, there is a psychological effect, and so what Mary's referring to is this first session for group B, where they actually start with the assistance of the device. And for something like detecting lesions, the device will help you find those lesions. And then, suddenly you're on the lookout for all these lesions. And then, once you hit the second half of of this first day, session 1, you're kind of on the hunt for these lesions, and you're really sensitive to these lesions, and yeah, that could change or affect the estimate of that reader's performance. It's hard to know, I guess. There maybe is some like post analysis you could do to show like if group A is different than group B. But yeah, I know which study you're talking about, and the FDA did really push back on making sure that this was done. They called it the variability with the day is. So yeah, session 1 for reader 1, maybe they didn't have a lot of coffee, but they did when they had session 2 or something. Yeah, it's supposed to help mitigate the effects of the variability for the specific day that the reading was done.
This idea of making sure, like the FDA not caring about, these sorts of effects is sort of related to another idea that pops up a lot in other like clinical trials. And it's a concept called intention to treat, which is where if you have a treatment that you perform and like the patient for whatever reason has to go to the other arm, that, or if they didn't have the treatment at first and then they got the treatment later, you actually still have to keep them and analyze them as if they never got the treatment. And so it's sort of like just a way of being very conservative so that if you do register an improvement, it really is real. Mm-hmm. Because you're sort of tying one of your hands behind your back and still showing a positive effect.
Yeah.
So it kind of rings to me like that.
Yeah, I want to make sure I get through some of these. I really only planned like half an hour for my slides, but we'll see if I can make it through them. The one method for establishing how a reader detects their lesions is as part of the experiment. So this is past the annotations for the ground truth. This is we're doing the experiments. You give a reader a case and they have to identify where the lesions are. One way to do that is just with a point annotation. You might need a software to facilitate that. So the reader will look at the case, provide a point annotation, and then for every point annotation, they associate a confidence. How confident they are that this lesion is a lesion, or that this mark represents a lesion. I know it looks like X to the 25th power, but that's X marks the spot and the confidence is 25. That's what this represents here. So yeah, the experiment will allow the readers to go through their cases and provide these annotations. These confidence scores are really important for the evaluation because I guess we'll go over it a little bit in another slide, but it's more so than just sensitivity, specificity. It's can you detect the lesions, and then how confident are you at detecting these lesions? And then once the experiment is done, they've made all their detections. The next step is to associate those detections with the ground truth that's already been established. I hope that shows up okay. But for the red lesion, that's the ground truth. The reader 1 found it and reader N found it. And then reader 1 and reader 2 also generated false positives. Typically, some software is written to capture those detections and associate them as true positives or false positives. And those will be captured in something like a spreadsheet that will be input for the analysis software. And that's what this might look like for this situation here. Yeah, long story short, just associating what the readers marked with the ground truth and determining if it's a true positive detection or a false positive detection, and making sure that they have a rating associated with their detection. I do want to talk about FROC curves. So FROC stands for free response ROC. I imagine quite a few of you are familiar with ROC curves. That they're similar to ROC curves. The top one here is an ROC curve. I know it's not square, but the diagonal is not where you want to be. That's the performance of a random guessing. And you want to be kind of up. You want your curve to go up towards this corner, this top left corner. Same thing with a FROC curve. The only difference really is we still have sensitivity as the y-axis, but the x-axes are different. For ROC, it's the specificity. And then for FROC, it's actually the false positives per image. And I'll go over that in a little bit. The nice thing about FROC is it does a better job at accounting for cases that may have multiple lesions. It captures those situations a little bit better, where ROC kind of squashes the data a little bit in terms of multiple lesions. It's not as equipped to evaluate those well. I don't know if you've seen the illustration of a dartboard. So for the situation on the left, you could throw a lot of darts at a dartboard and you're probably going to hit your bullseye. You're going to have a good sensitivity hitting that bullseye, but you're also going to generate a lot of false positives, where this situation on the right is going to be a much better situation where you're hitting your bullseye and maybe there's one false positive that's been generated. That's kind of what we're dealing with here. It's the same thing with ROC analysis, with FROC analysis. You want to be able to find your lesions without generating additional false positives. This is an example from one of our MRMC studies, and this is kind of what you're looking for at the end of your study. You have your line with the device and your line without the device, and there's a statistical analysis that can show that there's a significant difference between these lines. You want this line to be up closer to the top left corner. So that's a very good result. Yeah, I wanted to go over kind of the situations. We went over this with one of our clients to describe what constitutes a benefit of the device. The whole point of this study is to show that the readers are benefiting from using your device. So there's a bunch of different scenarios that your device will have an effect on the reader. So for these next images, I have a bunch of the situations. The image on the left is the reader's choice of when they are not influenced by the device, and then the one on the right is what they decided while using the device. So in this scenario, when they didn't use the device, they missed a lesion, and when they used the device, they found the lesion, true positive. And overall, that's a great thing. You improve the reader's sensitivity. So that's kind of like the standard. This is how you improve a reader, is by helping them find lesions that they would have normally missed. Then the opposite, which would be detrimental to your device performance, is they are able to find a lesion without using your device, and then for whatever reason, they use your device and they get worse. They don't find a lesion. That would be a bad thing. So you want to avoid those. But yeah, that's just kind of how the evaluation. What am I trying to say? When you're evaluating the device, this will be a detriment to your device. Another situation that's maybe not as obvious is improving the confidence of a true positive detection. So in the first situation, when they weren't using the device, they gave it a confidence of 85. However, when they use the device, they boosted their confidence. So they're much more certain that this is a lesion after using the device. And that is also a good thing for the evaluation. It shows that there is a positive effect of your device. So maybe not as obvious, but yeah, that's another thing that can happen. It's not just about finding the lesions, it's also about how confident you are at finding them. Another situation, false negatives. For either scenario, using the device, not using the device, they didn't find a lesion. That's actually kind of a neutral effect of your device. It's not good to miss lesions, obviously, but if you're not causing them to, if you're not the reason that you cause them to miss it, that's kind of not a big deal. It's just what it is. And another way that would be a bad thing that can happen a lot with devices is it generates more false positives, or causes a reader to generate more false positives. In the ground truth, there is no lesion. When they didn't use the device, they didn't find anything. But when they used the device for whatever reason, they thought they saw a lesion. Now, that would be a bad thing for your device's performance, and you hopefully can avoid those. But then the opposite is obviously a good thing. They generate a false positive when they weren't using the device. However, when using the device, they didn't put a detection there, and that would also be a good thing, a positive mark for your device. I think that might be, kind of last one, reducing the confidence of false positives. That would also be a good thing. While you don't want to be generating false positives, if you can, cause doubt on those false positives, that could also be considered a good thing as part of the evaluation. So yeah, I think that's all of them I had for there. Yeah, sometimes it's helpful to kind of go through that because it's not very obvious why we're doing all these detections and giving them scores. That's why I wanted to go through that, because it just paints a little bit better picture of what's going on and why we do this evaluation this way. And just keep in mind, this is just one reader reading one case. And for the study, we have multiple readers and multiple cases. So this is happening, many, many times over. So there's lots of data there. And then, yeah, at the end of the study, really you do your evaluation and hopefully get a nice positive results, statistically significant difference using your device versus not using the device. So yeah, sorry that explanation took a while. I think sometimes it's good to go into the weeds of this type of study. But I'll open up for questions and discussion. Did anyone have burning questions after going through the guidance and the protocol?
I have one. I don't know if it's burning, but I was wondering how statistical significance is determined. Is it just
Yeah, for FROC, it's, let me go back to, it's usually related to this curve. There is two curves that are generated, and you can calculate the area under those curves. And then there is a statistical method. I'll admit I do not know all the ins and outs of that statistical method, but there is a pretty well-defined statistical method that will evaluate your data to determine if this difference is significant. It accounts for the variability caused by the readers, both inter-reader variability and intra-reader variability, and also the variation associated with like the cases that were selected for the study. Yeah, it accounts for multiple sources of variation while determining this significance. Yeah.
I guess the main part of my question is it case by case or like FDA defined, or like depending on what your device does, like depending on the area of the body being scanned and that type of thing?
I mean, it's pretty device specific, I guess. I mean, this evaluation can be used for lots of different devices and lots of different studies. I'm not sure if that's what you're asking.
No, more so like the statistical significance, like say the brain scans, they would expect more significance. Scans of like the femur, they would expect less. Is that a thing? Or
Yeah, for at least for this curve that I'm showing right here, we did have to have a pre-sub with the FDA and say, we are expecting an improvement of 0.05. And there was a sample size estimation that helped us determine how many cases we needed to collect and how many readers that we should recruit. But that expected difference of 0.05 was really just, there's not necessarily a reason for choosing that number. It's often chosen. Really what that means is that the difference between the two curves, in this situation, the difference was 0.13. So it's much higher than 0.05. And that's great. That's why the p-value is so low. But yeah, I think that 0.05 is kind of like the standard chosen value for, a useful difference, I guess. Yeah, it's hard to know for sure. It is nice when you do your study and it's, it's much higher than that number, then you don't have to worry about it. But yeah, it seems like 0.05 is kind of this standard bump in the curve that you're looking for. For, like more critical things, I guess. I can't think of a situation that would be more critical, but you may want to have a larger boost in performance, and you'd have to establish that as part of your like sample size estimation.
Ethan, one question, like here you showed like one kind of lesion, right, or one segmentation of liver, of just one segmentation, like what is the size sample, like how many samples or how many images do you or FDA recommends for that? The second question would be like, if the device, instead of just doing one thing, is doing like multiple things, I don't know, like two more class A, two more class B, two more class C, like is the increase, I guess it would be like an increase of the size of the images that you need, but it will be linear, or so it will be like, okay, three types, you need 3 times the number of images, or it will be more, you need a biostatistician doing the work there and saying, no, you need whatever, like half of that because this or that.
Yeah, the first question I will answer, no, the FDA probably will not give you a recommendation for how many you should use. They really rely on the sponsor to justify how many cases that they're going to use. Yes, for the situation where you have like multiple things that you're detecting, I would definitely reach out to a biostatistician to determine how many cases you might need to do that. It just, adds another level of complexity. Yeah.
I've got a few things to add to that. So first, I would say there's definitely difference between having enough samples to prove statistical significance of some effect size. That's like one question, and usually the number that you need for that is a lot lower than the number you'll actually need to convince the FDA that your model generalizes. So the question as to whether the model will generalize is going to depend on the types of variability you might expect for that sort of device. So for example, if you have a device whose functionality might depend on body mass, then you'll definitely need to make sure you have enough samples of different types of body mass. Or if there's different types of lesions, like different disease states, you might need to cover that. Or an ethnicity is another one. So that's really common. In imaging studies, like different MRI scanner manufacturers is another. And so there can be kind of a long list and of sources of variability. And I would say in a lot of cases, it seems like 150 to 250 is like a pretty common kind of number you need of cases, but it really can vary a lot. And I think I would say we're hoping to bring someone on the team next year from FDA who will have a lot of experience with this, because they've done kind of research from the FDA side actually. Anyway, they'll be able to give probably a very good answer on that. But
That's awesome. Yeah, thank you.
And yeah, sorry, a lot of times we do get asked that question: how many cases do we need? How many readers do we need? And it's hard to it's hard to pull a number from out of your hat. Just say this is exactly what you need. If you want to be very, very thorough and you have unlimited time and resources, you would do a pilot study that actually uses your device on a small set of cases, maybe a few readers, and then you would estimate your variance based on that small study, and then you can predict how many you would need to do a bigger study. It's very difficult to do something like that. Yeah. And then going back to what David said, that may not necessarily cover the generalizability question. Like how does your device perform on these certain subgroups of data, like the imaging manufacturer, the different races in the patients, all that stuff. It really is just about the finding a statistically significant difference. So yeah, it's hard. Yeah.
Yeah. Got it.
And I would one other thing I'd just add in is definitely there's a difference between computer-aided detection and computer-aided diagnosis, and the numbers you need for diagnosis is much higher. And I think the numbers you need for like a screening application would be even higher. On the flip side, the numbers you need for like a triage application, like CAD-T, is lower. Is a general rule.
So I noticed that all these numbers, they include the device and the person. So is the device ever evaluated on its own? Let's say in some edge case, it produces wildly incorrect results, but the reader knows better and ignores whatever the device tells them and still reads, as good as they did without the device. So that's neutral, but the device might be completely wrong in some cases.
Yeah, that's a really good question, Ilya. The clinical performance assessment is one side of the evaluation. For every submission that I've been a part of, there's also standalone performance assessment, which is this performance of your device minus the human element. It's actually easier to perform. You just take the output of your device, compare it to your ground truth, like dice score for the lesion segmentations, and report that. That would detect, is my device way off and my readers are just really good. So yeah, it establishes what is this baseline performance of your device, how good is it detecting these lesions, and yeah, it removes that human element. So that is also included in a submission. I did have a small section related to it in that protocol that I created, but it does need a little bit of work to finish that out. But yeah, that's a good question. This is just really one side of the coin for the clinical performance assessment. There should also be an evaluation of the device itself.
And one hack that pops in my mind, like instead of having 3 reviewers, 3 kind of expensive radiologists, can you have like 2 per case? But instead of having only 2 radiologists, having like a pool of radiologists of 200 radiologists per hour, that kind of reduce some kind of bias, because you're putting more people there, but you're paying less because you have less. Yeah, you're paying less radiology hours. So is 3 a magic number for FDA or
3 is a good minimum usually. I mean, it can be used for tiebreakers or to establish a majority vote. Yeah, it does depend on what the task is. Like if for something like segmenting the liver, there may not be a lot of variability in outlining the liver. I mean, there's still some, but if you had 3 people do it versus 30, like you probably would end up with a very similar ground truth. When it comes to lesions, that may not be the case. Maybe 3 is definitely not enough to account for all that different variability. Maybe you do need 30 to come up with your ground truth. It's hard to know. There are like certain methods. I hope that they become accepted in the future where they have shown that a large group of essentially novices are as good as an expert if they're just given a little bit of training. And that you'd have like 20 people doing a segmentation and it's combined somehow, then that is as good sometimes as radiologists with 10 years of experience. I think there needs to be a little bit more evidence of that before that's accepted. But that would be perhaps another way that you could do your ground truthing is have a large pool of novices doing your segmentations and then combining them into a single ground truth.
I have a question about how much you're allowed to like rework things as you go, like once you get things going. Like it just seems like there's so many variables. Like I mean, just even like, oh, was the traffic bad on the way in to perform the reads? Like that could affect people's mindset and stuff. Are you allowed to, like, let's say you have a site and I don't know, there's some bad stuff going on in the world and people are just negative and it affects the reader results. Like, can you just throw out an entire site? Can you start over? Can you, what are you allowed to do once things get moving?
I'm going to defer to maybe Mary. Sorry, is Mary still here? Are you muted? Sorry.
I was muted and
Okay.
I'm still trying to figure things out. But anyway, yeah, so I think it kind of depends on the situation, of course. And I would find it a little bit hard because in real life, people are also going to be encountering those in real use. There's going to be other things impacting their performance as well. I guess if it's specifically relating to their performance when they're using the device, if there's an event you can justify, maybe. But I think in general, the FDA doesn't like you to trim your results based off of performance. And so I think you'd have to have a really, really good argument for that. And even then, I don't know if I've ever heard of the FDA allowing you to just cut out a particular site or data set for those reasons. But yeah, I mean, it's an interesting thought because I could see that being a factor, but I'd be nervous if that were the case.
That makes sense.
This isn't exactly related to what you're asking, but it's something I've seen a few times, and I don't think we've ever done this, or at least not that I'm aware of, is there'll be a pilot run of a number of practice cases, and you'll actually have the reviewers all do it, and then you'll have them cross-check each other and kind of debate out like, oh, here's why I did it this way and why I think it's better. And then you'll update the protocol for the annotations based on that. And then once it's kind of agreed on a small number of cases, then you'll actually run the study. And the purpose of that's to kind of improve get agreement on that protocol. I think I've just read about people doing that a number of times in other 510Ks and studies, but I don't know if we've ever done that. But I think too, I would say is if there's a sort of like site-specific variability or situation where you think the results would be unfavorable, it would definitely help you if you documented that in your protocol, and usually in the pre-sub you would kind of explain that, like here's how the criteria we would use to say this site isn't appropriate. I think this was on an IVD project that we weren't running the study or doing the strategy in at all, but I do think I recall there being something like that where there was a site where they kind of disqualified them, but I think it was like pre-written or something. Like I don't remember exactly. I just remember the client kind of complaining about the site and like, yeah, talking about it. So I might be misremembering because it's been a while, but
Yeah, if you can show that there was like a protocol deviation or that there was something about the study that wasn't followed properly, then that's like a legitimate reason why you could eliminate that. But the FDA, and I can't remember exactly where it is, but there is some like guidance or rule around withholding information that's pertinent to the that's pertinent to the review. And I think if you encountered a situation like that, I think it would probably be pertinent to review to discuss that and make sure that it was like an isolated case. But yeah, definitely would be case by case and concerning if that were to happen.
Yeah, well, thanks everybody. We are 1 minute over time, but yeah, I hope this was beneficial and hope everyone was able to get a little bit better understanding of these types of studies and why they're so important, why they're, a big deal to do. Yeah, and hope you have a good rest of your day.
Really great. Thanks,
Thank
Ethan.
you, Ethan. Thank you.
Thanks, Ethan. This is awesome.
Yeah.
Bye, everyone.